So neurons are probably the most important types of cell in the brain, and they are, as we'll learn, pretty complicated. But in a way, they're pretty simple because a neuron only really has three parts. You have the cell body, which is sometimes called the soma, or you might see it called the perikaryon, and then two types of neurites that we already talked about: the dendrites, which are again usually branched, and there can be a lot of them coming out of the soma, and then the axon. And there's only one axon in it, and then it can branch into multiple collaterals, but only one emerging from the cell body. And so again, these processes are unique to neurons. It's one of the things that makes them different from other types of cells. But in other ways, neurons are basically the same as pretty much every other cell. In fact, the cell body, for the most part, of a neuron is the same as any other cell in your body, and pretty much the same as any cell of any other eukaryote organism. So inside the cell body, or the soma, you have the nucleus, which of course contains the chromosomes, and then most of the inside of the cell body is made of this fluid called cytosol. Again, the cytosol of neurons is pretty much the same as the cytosol of other cells. It has a very high concentration of potassium ions, which will become important later on. And then a number of different organelles. And those organelles are just membrane-bound compartments that carry out the day-to-day functions of the cell. So the major ones are the ribosomes, the endoplasmic reticulum, the Golgi apparatus, and the mitochondria. So the nucleus, again, that's a structure that's found in every cell in your body, and every cell in your body contains inside the nucleus a set of structures called chromosomes. Those chromosomes, you probably know, are mostly just long stretches of DNA, and those chromosomes are divided into segments called genes, and genes are the instructions that tell the cell what kind of proteins to make, and the kind of proteins the cell makes determines what the cell does, because pretty much all the day-to-day functions, including metabolism and signaling and everything else that a cell does, is carried out by those proteins. So this process is the same in every cell in your body, and pretty much, again, every cell of every organism on Earth that we know of. So this process of gene expression, turning a gene into a protein, is called the central dogma. So again, the DNA, or the gene, is a DNA sequence that's part of a chromosome, and that gene gets transcribed into an RNA copy of one of the strands. So we call the first step transcription, and that RNA copy is going to carry the message in the gene to the cytoplasm, so we call it the messenger RNA. And then the messenger RNA leaves the nucleus, and then a ribosome translates the mRNA into a protein, and the amino acid sequence of the protein is determined by the nucleotide sequence of the mRNA. And again, this happens in every cell in your body, and in fact, every cell in your body has the same genes. So it doesn't matter if you're talking about a neuron or a liver cell or a skin cell, they all have the same genes, but only certain genes are expressed in certain cells. So there are some cells that are only expressed in neurons. There are some genes that are only expressed in neurons, and there are some genes that are only expressed in liver cells, and so on. Some are expressed in also, some genes are expressed in every cell, but some are unique. In fact, what determines the type of cell, or what determines what a cell turns into, so whether the precursor cell turns into a neuron or a liver cell or whatever, is the type of genes that are expressed. So the gene, in fact, is not just the coding sequence of a protein. Part of the gene also includes instructions that the cell follows that tells it when and where to express that gene. So, and those sequences are called promoters. So every gene includes a promoter and some other instructional sequences, and those promoter sequences are recognized by a special type of protein called transcription factors. And those transcription factors then are the ones that decide which genes get expressed in which cells. So again, neurons express genes expressed in neuron function, and so on and so forth. So the ribosome, though, is the structure that actually carries out the process of turning or creating proteins based on the RNA sequence. Ribosomes are found, again, not in the nucleus, so the mRNA has to leave the nucleus in order for translation to happen. And the ribosome simply reads along the messenger RNA from one end to another and makes a protein, one amino acid at a time, based on the sequence of bases in the RNA. Probably you've learned about this in some other biology class along the way, so hopefully this is a review for you. And you probably also know there are two types of ribosomes. There are free ribosomes, which means they're just kind of floating around in the cytosol. They might be in clusters of multiple ribosomes, but they're not attached to any membrane structure. And the proteins that get made by free ribosomes are ones that are just going to end up in the cytosol. So the proteins that don't get attached to the membrane just end up as cytosolic proteins or cytoplasmic proteins, and they're made by free ribosomes. And then the rest of the ribosomes are attached to another organelle called the endoplasmic reticulum. And in fact, endoplasmic reticulum that has ribosomes on it is called rough endoplasmic reticulum because if you look at it under the electron microscope, the ribosomes make little bumps on the surface. And those ribosomes make proteins who are destined to either be part of or embedded in the cell membrane or are going to be secreted out into the extracellular fluid. So those ribosomes sit on the ER membrane, and then as those proteins get made, they get sort of threaded through the membrane, either all the way in or they get embedded into that membrane. And in fact, this is important for neurons, because neurons have, as we'll learn, a lot of really important membrane proteins, things like neurotransmitter receptors and ion channels. And so neurons have a lot of rough ER, and that is mostly what you are looking at when you do a Nissl stain. So the Nissl stain, again, mostly sticks to nucleic acids, ribosomes, and mRNA. Both are made partly of nucleic acid, so ribosomes contain their own RNA as well. So when you're seeing the Nissl substance, that's what you're seeing. And the reason that the Nissl stain makes neurons, the cell bodies of neurons visible, is because their cell bodies contain lots of rough ER, making lots of membrane proteins. Whereas glial cells have less cytosol, and that cytosol contains less of the Nissl substance because, again, that's just not the way they work. So we'll be talking a lot more about membrane proteins later on. The Golgi apparatus is the other major membrane-bound organelle of the cell, and its function is mostly as the kind of the shipping department of the cell. So as membrane-bound proteins are synthesized on the rough ER, they get packaged into little vesicles. Those vesicles then fuse with the Golgi apparatus, and the proteins might go through some processing. They might get folded, they might get cleaved into smaller pieces, and eventually they get packaged into these little vesicles that then make their way to the cell membrane. And they're either going to get embedded in the cell membrane or get secreted out into the extracellular fluid. But that's kind of the main job of the Golgi apparatus. And it is named after Camille Golgi because he is the one that first identified them. And then there are the mitochondria. So you've probably heard the old saying that mitochondria are the powerhouse of the cell. And this is true in neurons just like it is in all other cells. So mitochondria, you probably know, take energy from food. So when you eat food, it's broken down into the major biological molecules, proteins, fats, and sugars. Those get broken down further in the cell into this stuff called pyruvic acid. And then pyruvic acid gets taken up by mitochondria. It goes through this reaction called the Krebs cycle. The energy from those molecules then gets used by the mitochondria to make ATP. ATP stands for adenosine triphosphate, and ATP is the main way that pretty much every reaction in your cell that requires energy gets its energy. So any reaction, any enzyme that needs energy will usually use a molecule of ATP to get that energy, and so that's produced in the mitochondria. Producing ATP also requires oxygen, so this is why you need to breathe oxygen, because oxygen is a small molecule. You can't really store it in high concentration, so you need a constant supply of oxygen specifically for this purpose. And then as it breaks these molecules down, the carbon gets converted into carbon dioxide, and then it also produces water. So the reason you need to exhale carbon dioxide is because it's the end product of this whole process. And this is especially important for neurons because, as we'll learn, neurons use a lot of energy. In fact, your brain uses more energy per weight than any other part of your body. The other major part of a neuron, and again, pretty much all cells, is the cytoskeleton. Cytoskeleton is kind of what it sounds like. The word cyto means cell, skeleton means skeleton. So this is kind of the main structural part of the inside of the cell. And the cytoskeleton consists of this network of proteins that give the cell its structure and its shape. It's important. These components are important for moving stuff around within the cell. Also, especially when cells are growing and developing and migrating to different parts of the brain, the cytoskeleton is important there as well. And there's three main types of fibers. You have microtubules, microfilaments, and neurofilaments, which in other kinds of cells are just called intermediate filaments. So microtubules are the biggest of the three. They're the thickest. They're made of these little subunits called tubulins. So one microtubule is a whole bunch of little tubulin proteins that form this coiled up polymer. And these are long, and they're especially important in the neurites. So these, you can see in the axon, these microtubules extend all the way down the axon. And so their job, among other things, is to help move stuff around the cell. So as stuff gets transported down the axon, microtubules act as kind of the highway for that. Microfilaments, meanwhile, are made of these little tiny subunits called actin. So actin, again, kind of like tubulin, is the individual proteins that make up microfilaments. And the microfilaments make up this kind of meshwork inside the cell, especially, again, also down the neurites, but then they're found along the inner surface of the membrane. So they act as sort of a structural component right underneath the membrane. The actin filaments or microfilaments are important for anchoring the cell into the extracellular matrix. Again, also really important for moving stuff around and remodeling the inside of the cell. And then neurofilaments are made of actually several different kinds of proteins, and they all have different functions depending on the cell type. Again, they mostly act as kind of a scaffolding, so all the organelles, usually when you see the inside of a cell, like a diagram, it kind of looks like everything's just sort of floating in empty space. But in reality, if you could see all these proteins, what you would see is all these organelles are really just embedded in this matrix of neurofilament proteins, and they're all attached to those proteins with various adapter molecules. So we'll come back and talk about some of these later on, probably. So probably the most specialized part of the neuron is the axon. So the axon is typically, for most cells or most neurons, is a pretty long neurite. So it extends from the cell body up to a meter in length. So a meter is a pretty long distance for a cell. In fact, there is no other single cellular structure that is that long, and that's of course in humans. If you look at larger animals like whales and elephants, their axons can be especially the ones in the peripheral nervous system, can be several meters in length. And so each axon comes out, and then they're typically pretty thin too. So even though they're very, very long, they're usually thin. Although there can be axons that are up to 1 millimeter in thickness, and we'll talk later about how the thickness or the diameter of an axon affects its function. The axon, by definition, emerges from the cell body at a location called the axon hillock. And all neurons, or at least all neurons that have been identified, only have one axon that emerges from one axon hillock. Now, that axon can branch after it leaves the axon hillock. So even though there's only one connection to the cell body, the axon itself can eventually form many, many connections, as long as it can form more collaterals. And as we'll learn later, when the signal called the action potential is generated at the axon hillock, it travels usually away from the cell body, and then when it hits those axon collaterals, those branches, it splits off and goes to all of them, eventually reaching the terminal. Now, the axon, one thing that makes the axon different from the rest of the cell is that inside the axon, the cytoplasm does not contain any ribosomes or rough endoplasmic reticulum. And what that means is that all the proteins, everything inside the axon that needs to get made, is made in the cell body and then has to be transported down the axon. So that process of moving stuff down the axon is called axoplasmic transport. And for the most part, it's carried out by these special motor proteins. And the reason that you need these special motor proteins is that, again, axons can be very, very long. And diffusion, the sort of natural tendency for molecules to move from an area of high concentration to low concentration, is slow. So if you had to wait, for example, for a protein that got synthesized in the cell body to diffuse all the way down the axon, again, let's say it's an axon going down to your leg, it could take a matter of days before those molecules got to the end of the axon. So axoplasmic transport is a faster way of moving stuff down the axon. It does require energy, and it requires these little motor proteins. So there are two types of axoplasmic transport. Anything going from the cell body or away from the cell body down toward the axon terminal, we call anterograde. So this is anterograde transport. And then anything that is moved from the axon terminal back to the cell body we call retrograde. And they both require these motor proteins. The forward anterograde motion is carried out by a protein called kinesin. Retrograde motion is carried out by another one called dynein. Again, they both use energy, and they both attach to the microtubules and then to vesicles, which carry, again, maybe membrane proteins or other things that might be needed at the terminal of the axon. And they essentially sort of walk down the microtubule. So they have these little binding sites that attach to the microtubule, and then when they use ATP, they release from the microtubule on one of those ends, and then that end rotates forward and grabs on, and they repeat that process. So it actually kind of looks like this. This is like just an animation of kind of what it might look like for kinesin protein to be walking down a microtubule. Here's the microtubule down here. Again, this whole thing is a big giant vesicle, and you can see little proteins embedded in the membrane there. And then this little guy walking along would be the kinesin protein. So every time it takes a step, it's using ATP and rotating and releasing and then rebinding to the microtubule. And so this is how most large molecules end up at the end of the axon. And then the very end of the axon, so eventually if you trace an axon long enough, you will get to the axon terminal, which is the first half of a structure called the synapse. So the synapse is the place where one neuron connects to another neuron, or sometimes to other types of cells. And again, Ramonica Hall showed or theorized that these were the contact points between cells. where they make physical contact, but they don't actually fuse together. And the end of the axon we call the presynaptic terminal, because generally, as we'll learn more about later, information flow goes from the presynaptic terminal, again, which is the end of the axon, to whatever the postsynaptic cell is. So postsynaptic side could be a dendrite, it could be a cell body, could be another type of cell altogether. But again, the presynaptic terminal is the end of the axon. It's a little bit different from the rest of the axon. And for one thing, there are no microtubules that extend into the terminal. There are lots of mitochondria, so the process of synaptic communication requires energy. And then the most important part of the presynaptic terminal are these little bubbles of membrane called synaptic vesicles. So a synaptic vesicle, these synaptic vesicles contain neurotransmitters. So when one neuron communicates with another, these synaptic vesicles kind of fuse with the membrane, and they diffuse into this space here between the cells called the synaptic cleft. And then they travel across the synaptic cleft, the neurotransmitters, and they bind to these proteins that should be on the membrane of the postsynaptic cell, which again can be another neuron, could be a muscle, just depends on the type of cell, type of neuron. And then the receptors on the postsynaptic membrane then trigger some sort of response, intracellular response in the postsynaptic cell, and that's how neurons communicate with each other. So we'll learn later about how the signal gets here in the first place. So you'll have an electrical signal called an action potential that gets generated at the axon hillock. That action potential travels all the way down the axon very, very quickly. And then eventually, so again, this represents the action potential. And that electrical signal, when it reaches the presynaptic terminal, it triggers the release of the synaptic vesicles. And this is generally the direction of informational flow in neurons. So from the presynaptic terminal to the postsynaptic terminal, or postsynaptic membrane. And there are lots of different types of synapses, like I said. So you can have synapses, mostly we'll be talking about later on, we'll be talking about synapses between neurons, but you can also have a synapse from a neuron to, for example, a muscle cell. And the synapse always starts, the presynaptic terminal is always the end of an axon, but the postsynaptic membrane can be on the cell body of a neuron, it can be on the dendrite. So if the synapse is between an axon and the dendrite of a neuron, we call it axodendritic. So this is an axodendritic synapse. If the synapse forms on the cell body or the soma of another neuron, we call it axosomatic. This is an axosomatic synapse. And you can even have synapses between axons or between an axon and another axon. So in this case, the presynaptic terminal forms a synapse with the shaft of another axon of another neuron. So that's called an axoaxonic synapse. You can even have synapses where the neuron forms a synapse with itself. So you can have a case like this where a collateral can come back and form a synapse back with the cell body of the neuron it came from. This is called a recurrent synapse. And then the dendrites are the other important specialization of neurons. So both axons and dendrites are generally unique to neurons. No other cells really have these structures. Dendrites are different from axons in that they're usually shorter. They don't extend very far away from the cell body. And unlike axons, a neuron can have lots of dendrites or neurites that become dendrites that come out of the cell body. So again, an axon, there's only one axon or one axon hillock per neuron, but you can have lots of dendrites coming out of the same neuron. And then those dendrites can branch. So in fact, this is where the word dendrite comes from. It comes from, I think, the Greek word for tree. And sometimes you'll even see or hear the dendritic structures called dendritic arbors, which is, again, just means tree. And again, axons can branch also, but dendrites, again, because they're shorter, they just appear much more branched than axons. Some of them can be spiny, so you can see dendrites with little bumps along the shaft. Those are usually the sites of special kinds of synapses. It's probably a way for the cell to sort of compartmentalize the components, the specialized postsynaptic molecular components that are needed there. And unlike axons, ribosomes can contain, dendrites can contain free ribosomes. So inside the shaft, you can see ribosomes. So the proteins that a dendrite needs can be synthesized kind of on site. So you still can have active transport moving stuff along the dendrite, but some proteins are synthesized right at the inside the dendrite where they're used. So next time we will talk about the different types of neurons and how we classify them.